now, it's brought us at the brink of a genetic revolution where it goes like on one end of the spectrum there are changes that we can do to see what are the natural phenomena that are occurring, which are still not unveiled currently by the technologies. And on the other hand, we can imagine certain uh, edited crops or certain important products through this technology that can help us to safeguard our health, and they can also introduce certain important mutations that can revert or recover certain genetic disorders that are specifically based on a single mutation or a single base mutation. They can fight the infectious, we can do that for the infectious diseases also. We can develop diagnostic kits, like we can create animal models to study human diseases, which are, I hear some noise in the background. Is it because of me? It's really disturbing. So kindly please reduce the noise in the background. I'm sorry. And in addition, we could also use these innovative technologies to create new biofuels, new greener chemicals for industries, using these technologies to develop altered microbes that can give us industrial enzymes or industrial chemicals. For example, we have in ICGEB, people in biofuels are developing certain, modifying, introducing the butanol, anti-butanol pathway, butanol acidic pathway into the E. coli to develop new biofuels. So this is like poised to salvage our food supply, climate change, and to prevent plants against competition from weeds. Also increase the nutrient content in our grains and in our green vegetables through, and also prevent pathogen infections. So this is like not new. Nature has been editing genomes for a very long time, for thousands of years, with natural variants which are, they are natural variants that are being developed through natural selection, both in animals and plants, with which have enabled the genomic variants for these organisms to serve. Moreover, human beings have been, employing artificial selection to domesticate crops for more than 10,000 years. The quantum of crops that we presently consume have been domesticated from their wild ancestors, from their wild progenitors or ancestors in the last 12,000 years. And this has been the case for in the maize, where the wild ancestor Teosinte gave rise through domestication process to the current hybrid corn that we have. So the current technologies that are present, that are being used by human beings for artificially selecting, are crossbreeding, mutation breeding, and transgeneration. So these crossbreeding and mutation breeding have been the major contributors to the green revolution that we have seen. Basically, crossbreeding is the main as technique that has given us new varieties of hybrid varieties, dwarf varieties that have increased yield. So this crop breeding basically relies mainly on introgression of existing natural genetic variation into elite backgrounds, which requires substantial germ plasm, resources that are there, but we have not yet utilized it. And it requires further, once you cross them, then you require extensive back crossing followed by selection of progeny with for screening for the best the genotypes for the best agronomic traits, and this is time consuming, labor intensive. However, further mutation breeding also is like the nature is or is stochastic. I mean, it is limited by this stochastic nature because it's random and it can occur throughout the genome, which may lead to unnecessary chromosomal rearrangements or some effects that are not required. And also crossbreeding has two disadvantages too, because we need to cross the plants a father and a mother together to produce the hybrid F1 generation, and then that process to stabilize the new trait. However, this phenotypic variation that is present is restricted in this case because we cannot cross two very different or two very different genomes. We need to cross those which are close to each other. They have close relationship. Thereby, we cannot introduce innovative mutations into the plant. However, and then transgenic breeding was there, which is being, let down by several countries because they do not like to eat these GMOs. However, eventually these have led to development of new varieties, but they have not yet been accepted by the scientific regulations. So the newer technology, which is supposed to be the genome editing, which is not just genome editing, but it's known as a precision design agriculture, which is involved in developing these new edited lines or are involved in introducing mutations, thereby introducing an important trait in the crops, is not just alone, but in consonance or in integration with the functional genomics strategies that are available, with precision editing technologies that are available, and also synergizing the big data analysis into the development of these crops. So, as I said already, this domestication process has given us the current amount of plant varieties that are being used now. However, this process takes a lot of time, thousands of years, and this genetic diversity eventually is still present there. But we have no information about how much is there and what all we can use. And this takes a lot of time. But in this case, in case of genome editing, in addition to breeding, we can supplement this to breeding by introducing an event or a specific mutation, which may then be crossed an elite background through crossbreeding, and then we can do field evaluation of this, and this would take less time, not thousands of years. So this is what we envision through genome editing to reduce the domestication time of these crops, and then eventually speed up the selection and domestication of elite wild plants, and thereby utilize the genetic diversity that is available, as well as introduce or inculcate this genetic diversity through genome editing tools. So as we've seen that in plants, these wild crops which are there, I mean, this de novo domestication has been seen through in the past few years. We've seen this editing technologies have been used to domesticate tomato, a species Solanum pimpinellifolia. It's been domesticated by targeting these 5 genes to increase the fruit size, the fruit number, as well as the lycopene content of the fruit. Similarly, the rice has been domesticated through genome editing by working upon these genes that are mentioned earlier. Some of them are QGS, quantitative trait loci, which may affect several genes together. However, these have been tried to being employed through gene knockouts and QTL knockouts, and they have seen that it has influenced the tillering number, the architecture of the plant, the grain size, the grain weight, the number of grains produced, and also the on length, as well as shatterproof trait in certain wild rice. For example, it's polyploidy rice that is present in the African country. Some African countries do have this rice, and this has been used. And then it can also be used for creating, hybrids. For example, in every time thousands of hybrids are being used every year by these companies, and the vigor generally decreases after the first F1, and we need to maintain that F1 hybrid every time we need to cross and create it every year, and the farmers have to buy these seeds every year. So so through genome editing, we could fix this F1 vigor, or the hybrid vigor in the F1 filial generation by targeting the meiotic and the fertilization genes using this technique of CRISPR editing, and try to restrict the meiosis and convert the meiosis into complete mitosis, thereby eliminating any gamete. Specifically, people are trying to use androgenesis, but they can be gynogenesis also, or parthenogenesis also. But this technique which is being used is generally being targeting the enzymes in the pollen grains, and these are somewhat like pattern-like phospholipase C, and they have a specific role in the membrane development. And sometimes when when these are knocked out through mutation, mutagenesis, it can lead to creation of this male defunctional, or male paternal chromosome gets degraded. Thereby, you can use this line for creating hybrid vigor. So similarly, there are several genes that have been identified to create haploid induction. So generally, haploids are basically developed for making new inbred lines, and these new inbred lines are then further doubled through colchicine, and this doubling then leads to increased haploid induction, or double haploid induction leads to agronomic better agronomic performances. Hence, these have been also identified, and this technique of genome editing has been used to induce this double haploidy, also to introduce qualities like higher yield and enhanced resilience to stress and quality, improved quality. Similarly, for developing the modified crops which have a complete pathway inside, for example, I said the butanol pathway or the photosynthetic pathway have been introduced in seed crops to increase the photosynthetic efficiency. Similarly, CRISPR has been used to identify certain genes, where modify certain genes in the microbes like bacteria, algae, fungi, that are important for industrial uses, for developing certain enzymes for industrial uses, and these can then be used for producing green chemicals, including biofuels and biomaterials. So this is a slide that is showing the breeding, plant breeding milestones here, and you could see that around 12,000 BC, you can see that the initial plant breeding started, and the domestication began through there, and then eventually the first plant that was domesticated was emerald wheat, and eventually then the totipotency tissue culture techniques also started upon, and it was first demonstrated in carrot, and then further the first the crossbreeding techniques were used to develop these important varieties of crops, and eventually in around 2000 and late 20th century, the transgenic lines came up, and eventually in 2019 the first genome edited crop came up, which was basically the high oleic acid soybean, which was developed for cooking stability. The oleic acid increased oleic acid, or omega-9 increases the cooking stability of the soybean oil. So these have been the timelines of this, and then you could see the different traits that have been introduced through genome editing. So on the first upper portion, you can see how the different traits that like herbicide resistance, and blight resistance, powdery mildew resistance in wheat, waxy corn, wherein it is, myelopectin has been increased for industrial uses, and soybean with reduced trans fats, non-browning or anti-browning mushrooms. So there are several of them, gluten-free wheat, etc. And they've also, the technique has been also been used to fight the human diseases like leukemia, HIV, lung cancer. But however, these are not yet being practiced due to some ethical issues that we need to look upon. But yeah, in cattle and all, they have been used for improving the diseases, for disease resistance. And here you can see the list of genes that are being utilized in the important crops that we generally use on the daily basis. For blight, we use the sweet genes that are being mutated for increasing blight resistance. For blast, they use this ethylene responsive factor 922, which has somehow although being a quantitative trait, has it has a polygenic influence. However, still this transcription factor, when mutated, led to the resistance of increased resistance to blast. There are several of these here available. You can check the literature out if you need to look into the details. So previously, like, mutations were used in artificial induction of artificial mutations through chemical mutagens as well as ionizing radiations. Even plants EMS, ethyl methane sulfonate, is being used, and these are the two scientists that were who had pioneered to introduce such mutations. Not they've done it in Drosophila, but they have been used. These have been used in plant developing plant mutants also. However, the technique before the CRISPR came up, the main scientist who actually introduced this strategy of gene targeting, which later then was used as a genome editing tool, was Mario Capicchi, and he was basically a PhD from the University of Utah, and along with the other two scientists, he received the Nobel Prize in 27, and his work, Nobel Prize, recognized the pioneering work that he did for developing different knockout mice models for utilizing them in the labs to test our different drugs or different strategies to deal with diseases. So without these animal models, we cannot do our testings, or we cannot test the different whatever strategies that we employ. So he's the one who utilized this, and this he developed through he tried to introduce double selection, a positive and a negative selection, for selecting or enriching the cells or events or the embryonic stem cells which have the edited mutation. So he was the first one to introduce this technique of gene targeting. And later on, these two scientists, Emmanuel M. Charpentier and Jennifer Doudna, were given a Nobel Prize for first proposing the CRISPR-Cas9 enzymes that had been can have a combination of Cas9 as well as a guide sequence together in consonance, can be repurposed or re-engineered to mutate or create double-strand breaks in genomes, and thereby create mutations for introducing several important traits. So this is a slide where you can see that when the initial this discovery of CRISPR came out through a very simple experiment, a simple project of how viruses affect bacteria. And here you could see that the adaptive immunity of bacteria, wherein the foreign DNA from the viruses, when the virus attacks initially, is a piece of the viral genome is put into this is acquired into this CRISPR loci, wherein it is then transcribed, and once it is transcribed, it leads to the production of this tracer RNA, the transcribed CRISPR RNA, and along with this CRISPR RNA, the tracer RNA binds and forms the guide RNA together, to which directs the Cas9 to create a double-strand break. So it's like a piece that is included is like every time a virus infects, a piece of DNA from the virus is introduced into the bacterial genome, and hence this seems like a genetic vaccination card of the bacteria, where different previous infections of the virus are noted down, and whenever the virus again comes and attacks the bacteria, it identifies through this piece, the RNA DNA complementarity happens, and then the Cas9 is directed to create a double strand break in the viral genome and thereby desensitize or kill the virus, and thereby protecting the bacteria. So in connection to this, several other endonucleases, homing endonucleases, and several other endonucleases had been used. Site-directed endonucleases were used, but CRISPR was found out to be really simple and easy to develop. The primers were easy to develop, and it was simpler than the meganucleases, zinc meganucleases, and talent, which required elaborate protein engineering to identify the DNA motifs for every specific sequence. So it was time consuming and costly, and hence CRISPR came up as a redeemer, and it allowed us to reduce the time and the cost involved in this process. So these programmable, all these endonucleases that are used previously, and including the Cas9, they all create double strand break, specifically because the Cas9 has two domains that create the double strand breaks. The RUVC domain creates a break at the non-complementary strand, whereas the HNH domain in the Cas9 creates the break at the complementary strand. So there are different versions of it now available. People mutate one of these and then use that platform as a nickase, wherein only a single nick is made on the DNA and no double strand breaks away. So I would be showing that in the future slides. So this is how a double strand break is created from either of these endonucleases, and eventually the endogenous repair machinery, it repairs this break. It identifies this break and it repairs this break, and this is the source of introduction of mutations in the genome. So either the repair can be through homologous non-homologous end joining or through homology-directed DNA repair system. However, this HDR, or the homology-directed repair system, is more precise than the non-homologous one, because this is merely insertions or deletions occurring here, which disrupts the gene or introduces a stop codon in the coding sequence, thereby the protein is not completely formed, and hence the function is totally lost. Thereby we say that the gene has been knocked out. And in this case, the homology-directed repair has been used to develop the desired mutations in a protein or in a gene through sending or providing a homology donor repair template, which is along with the Cas9 and the guide RNA. This template has to be given, where it utilizes the the scientific whatever this is, this homologous recombination, site-specific homologous recombination strategy to introduce the desired mutation, because it finds the left and the right arm, which is homologies are there, and whatever is there between the homologies is transferred into the genome. So this is how different genome editing tools have been now developed, and besides Cas9, there is CPF1, and then there is Cas13 nuclease also. Then several modifications of these Cas9 proteins have also been done by introducing sorry, I'm sorry for this one second. So the cytosine base editors and adenine base editors are nothing but basically these are using platforms of nicases or dead Cas9s, where the Cas9 either both the the domains are mutated, or one of the domains are mutated. For example, here you can see that the HNH is working and the RUVC domain is mutated through a mutation at D10A, and this finally it leads to probably a platform wherein you can tag in another domain, like a cytidine deaminase or an adenine deaminase, which along with the Cas9, the Cas9 tries to utilize this function of these minases to convert point mutations, create point mutations like cytidine deaminase. Deaminase is the cytosine to thymine, whereas the adenine base editors, deaminase adenine to guanine. So this A to G and this is C to T. So these help in introducing point mutations. Then comes up the final, the latest one is the prime editing, where you do not create any double strand breaks, but you just introduce the template or the sequence that you wish to be introduced is introduced along with the primer binding site, and there is a reverse transcriptase that is sitting along with the nickase there inside, and that it reads this template that you've sent and it creates its own inside the cell. Thereby, you do not require any double strand breaks to be created, so less off target. So this is these are the different. It's a summary of the different enzymes and endonucleases that have been used. The latest one is the Cas14 that has come up. So the Cas9 uses double strand DNA as a substrate, the CPF1-2 along with that, and Cas13A is used for RNA editing. Single strand RNA is a substrate for this, and the 14 is also a single strand. It's a newer DNA endonuclease. Recently, it has been identified, and this cuts single strand DNA. So these are the different binding domains that are active here. So the Cas9 has both the domains active, and the CPF1 has just one domain. Originally, therefore, it is slightly smaller than the Cas9, so easier to introduce in the cells. And the Cas13 and 14 have different. RUVC is the same as the Cas9 one. However, they are smaller. They are smaller enzymes or proteins than the Cas9. So they have different flexibilities. They have their own pros and cons. So depending upon the experiment, we need to choose. So whatever present as of now are type 2 and type 5. So there are two classes of, majorly two classes of these CRISPR endonucleases, CRISPR endonucleases, and in each class there are three subtypes, so total 6 subtypes are there eventually, and there are many subtypes there, 33 further divisions are there. However, the major ones that are used in plant genome engineering are the type 2, that is the Cas9, as well as the type 5, that is the CPF1 or 12A. So these are the two that are being used eventually for plant genome editing, simply because of the simple nature of editing as well as multiplexing. So they can be easily used for multiplexing, stacking 2 or more guide RNAs, or more than 2-4 guide RNAs, 2-3-4 guide RNAs, or more than that can be multiplexed through and used for this. So simple the ease of performing multiplexing. So these are the two important ones. So this is how the Cas9 cuts and creates a double blunt end, and the CPF1 creates a staggered end. And eventually, they say it is the blunt end is easier for the non-NEH-EJ pathway repair. It goes towards it, and the staggered end promotes homology donor repair. However, it is not strictly followed. We can introduce differences too, and hence these have been utilized eventually for. So the selection now. The important thing that we now to look into the experiment is how to perform the experiment in the lab. So in that case, we need to select the target genes, and an appropriate genome editing approach has also to be utilized, as there are umpteen now tools available for genome editing, and the approaches are also different. So one has to see how to go about that. So in the case of the experiment that we need to work upon is, first of all, we need to have the whole genome sequence in our hand. So this whole genome sequence is required because when we are about to design the guide RNAs for developing the constructs, so that the guide RNA needs to be verified for any undue off targets. So it needs to be specific and work on target and specific. So for that we put the sequence of the target sequence, we put it in the softwares that are available, and these softwares then help us to screen the best guide RNA. So this is one of the way you need to check whether your guide RNAs are specific and they are not creating any off targets in the other genes, and they are not creating double strand breaks or nicks in the other parts of the genome. So for this, and then one of the crucial issues to be considered would be the choice of the gene. So once you see when you are talking about editing single genes, it is easier and more doable. For example, some traits like grain amylose content, and in the rice you see grain amylose content, or if you see the oil quality in the soybean. So these are controlled by single genes. So genes that are controlled by single genes are the most appropriate ones to go ahead eventually, because they are not affecting any other aspects or developmental processes. So on the other hand, the crucial agronomic traits, for example, the traits that are concerned with quantitative traits like yield and abiotic, biotic stress tolerance, they qualify the quantitative category, and thus they are regulated by QTLs, quantitative trait loci, and many of which are, in the recent past, they have been identified, they are mapped, and eventually cloned. However, they are a huge bunch of them there, and they are offer an attractive source of potential targets for genome editing plants. But you need to work with a pinch of salt, because they may affect other developmental processes, because they may be involved in some other sub processes. So this need to be taken up with some caution. And this is now the slide wherein I would show you the first important strategy with employing this tool of genome editing is for gain or loss of function mutations. So firstly, you can create frame shift mutations or generate loss of function alleles. You can create amino acid substitutions through these tools. For example, you can use ABs and CBs, editing base editors, cycling base editors for creating partial loss of function or gain of function, which eventually leads to different allelic expressions of these genes. Then you can also use these strategies for screening key functional sites in a protein. For example, some unknown functional sites there, which we do not have, we have not yet characterized. So these can be characterized through these. So you can create a guide RNA array of the entire genome and put the entire genome guide RNAs into a library, and these library guide RNA library then can be as a whole pooled and transformed through agrobacterium method into this plant, and this plant can be introduced in the embryos, in the callus or protoplasm, whichever form, generally callus embryos, and then these callus can be regenerated and genotyped. Mutations then can be screened through sequencing or whole genome sequencing or many other screening other processes. Genotypically also to identify the beneficial elements. So here we can see the examples of these gain or loss of mutation. You can see this rice, where high amylose rice has been developed through this technology, because we need this as these high amylose content or resistant rice. Resistant starch is important for diabetic patients, because it does not degrade fast and does not give sugar spike. So people have tried to mutate these starch branching enzyme to the VAXI gene. The VAXI gene they have tried to retain, and knock out the gene that is creating the amylopectin, so that we have a lot of amylose. Instead of amylopectin, we convert the entire sucrose into the amylose here. So this is how they've increased this amylose here, and they try to increase the oleic acid here in soybean. They try to increase the amylopectin in corn, that is the waxy corn. In this case, they have knocked out this gene here, the waxy gene, so that the sucrose is completely converted into amylopectin and used for industrial uses. And then also we have here rice with enhanced fragrance, for example. The gene that is the betaine aldehyde dehydrogenase has been knocked out, which actually converts this GABA aminobutyraldehyde into GABA. And instead, the entire gamma aminobutyraldehyde is directed towards synthesis of 2-acetylpyrroline, which is giving the fragrance in rice. So similarly, this thing has been done in maize also. So these are, then there are glutinous, similar glutinous waxy rice varieties have been developed by knocking out the waxy gene and creating more sticky or glutinous rice varieties for eating through using chopsticks to eat them. So people require that, consumers require that character of glutinous trait. Similarly, there are tomatoes that have been, developed with increased vitamin D as a source, because the main source of vitamin D generally we say is sunlight. However, we human beings, have this conversion in themselves. They absorb this sunlight and create vitamin D. However, this is not sufficient enough. So people have, two groups have tried to knock out these two genes that are involved in the cholesterol biosynthetic pathway, and which ultimately leads to synthesis of provitamin B, which eventually in the presence of sunlight leads to production of vitamin D3. So these two groups have identified these two genes, DR1, DR2 and DR1. They have been knocked out, and they try to increase the vitamin D in the tomato fruit. So here you can see that the pathway here, starting with the acetyl coenzyme A. So the end product is generally cholesterol here through 7-DR2 gene, and the other 7-DR1 is creating the brasnolide and the sigma-sterol. So these two important groups of compounds here, they are important for the plant, but brasnolide is basically helping in cell expansion and cell division, and also helps in etiolation and reproduction. And the other stigmasterols, these are also important for pathogen resistance. So what they have done, try to is they've knocked out the 7-dehydro cholesterol gene, thereby the entire acetyl coenzyme A is going towards the synthesis of this product, the provitamin A, which in the presence of UVB leads to the production of vitamin D3. So this is the experiment they've done. They've used these, they've shown how they designed the guide RNAs, the two guide RNAs they've designed. This is the target sequences, and this is the PAM sequence that is there here. Sorry, the PAM sequence here, and here these two red regions. And they have tried then sequence after knocking out, they've seen sequence and seen that a huge chunk of the DNA is lost, thereby creating a mutation, or the frameshift mutation. And here they've shown that how the hydrocholesterol, this is it's completely increasing here in the mutants. And this is in the presence of sunlight creates provitamin D. So this is how we have shown. So then the other technique is that CRISPR mediated genome editing in plants by loss of gain function that I've told, and now here we are trying to use it in different ways. For example, we have tried to increase the grain weight of the cereals, the grain size, the number of grains have been improved, the length of the grain has been increased, and then the fragrance of the grains have been tweaked. and the chalkiness has been tweaked, and then iron and zinc content has been enhanced. So all these have been tried through genome editing. Here already previously I showed you the blast resistance that has been introduced through editing of this transcription factor, ethylene responsive transcription factor, and then they've also used it for studying, for basic, knowing the basic phenomena and the basic mechanisms that are affecting flowering, because inflorescence architecture is the main trait that leads to the controlling of the yield. So here they have in Arabidopsis and also in other plants, they've tried to mutate these important genes that affect flowering. So these are all genes that are affecting flowering development in some way or the other. This is the apetala one, the short vegetative phase gene, and the terminal flowering locus one. So they have individually knocked them out and seen how it behaves, and how it eventually, when all the three mutations are triple mutant is seen, and how it is behaving. So this is in general to know the phenomena, how this is working. So we can use for basic studies also, you can use this technique. Then here the other one is where how you have to increase the herbicide resistance, for example. In this case, astrolactate synthase is the enzyme which is important for the synthesis of branched chain amino acids that are essential amino acids. And if the inhibitors of this enzyme are being used as herbicides, and they are used for killing weeds. However, when you spray the plant with this herbicide, your main crop also is affected. So to protect your main crop, people have tried to introduce these mutations, these two mutations, this tryptophan to lysine and serine to isoleucine at these points. And this is through homology donor template, to give a template with the right arm and left arm homologies, and with the help of site-specific homologous recombination, this central portion with the desired template is shot inside, and then these are sequenced, and the mutations are then verified, and you can see the phenotype also when you spray the sulfonyl urea or chlorosulfuron or bispirax sodium. These plants are tolerating or resistant to these herbicides. When you're spraying the herbicide, the weed would, the control plant dies, but the edited plants is surviving happily. Similarly, there are certain variants. For example, in this case, Argos 8 is supposed to be a negative regulator of ethylene responses, and Argos 8, generally people have used for transgenic development, and they have seen that it increases drought tolerance. But somehow when they screened the 400 maize inbred lines, they found that the expression of this Argos 8 was very less. So they developed different variants of Argos 8 by swapping the promoter of this Argos 8 with a GOS2 promoter. Is a highly expressing constitutive promoter. So they increased the expression of this Argos 8 genes, and different variants were developed. And this they showed in the field that it really increased the flowering in the grain yield as well as the drought stress tolerance. The base editors are definitely an upgraded form tool of the CRISPR-Cas9, wherein the platform is basically a nickase, where one of the domains of the Cas9 is mutated, and only one domain is active, which can create a nick. So how it works is like, if you use a deaminase domain along with the nickase platform, the deaminates the cytidine to uridine, and this uridine then and uridine along with the guanine is recognized as a mismatch in the cell, and this mismatch is then taken up, and this non-targeted, non-edited strand is then nicked by the endogenous repair mechanism in the cell, and this guanine is then converted to adenine, and then with a further replication step, this transforms into the thymine. So eventually it's rectified. So a C is transformed into a T. However, sometimes when you use this, the cytidine is deaminated to uridine. but there are certain enzymes that convert this uridine back to cytidine. There are enzymes there, so we need to use the inhibitors of those enzymes so that this uridine is not converted back to cytidine. So there are those things you have to take care of. So all this, eventually David Liu's lab had developed these editors, but they were not very happy with this editing technology because they created several off targets at the RNA level and at the DNA level. So they tried to upgrade this technology by introducing the prime editing technology, wherein you can see you can see the video. Probably if it is able to. I think it's not able to play the video, but the video showed that the four mutations, point mutations could occur. So like the CRISPR-Cas9 behaved like a scissor, molecular scissor, creating double strand break. The base editors work like a molecular pencil, creating point mutations, and the prime editing technology is like creating all the 12 transversions and transitions together. It's not just C to T and A to G, but it is like C to T, T to A to G, any or whatever possible transversions or transitions that are available can be done through this simple technique of editing. And you require a PEG RNA. Instead of the guide RNA, you just require a prime editing RNA where it comprises the binding site for a primer as well as a template that is harbored, which is the edit for the for being utilized by the reverse transcriptase. It reads this edit and it develops the complementary strand. So this is how a Nikkei's platform allows the a Nikkei's platform along with a reverse transcriptase is the latest technology which people are using for editing crops with high speed and less off-target effects. So this is how it is done. That initially, this Nikkei's and the reverse transcriptase are introduced, and the PEG RNA is also introduced along with this, which is supposedly having the binding site as well as the edited sequence, which is used as a template by the reverse transcriptase to read. So it reads the RNA letters here, and it attaches the corresponding sequence along with it. And the old sequence, the original sequence, is excised through the endogenous repair, and now you have a mismatch here. This mismatch is read by the endogenous repair mechanism. Again, the nick is created to remove the unedited strand. So after removal of this unedited strand, the new strand is taken as the base or the template for the RT to develop the complementary strand too. So hence you get a fully edited DNA in this. So this is one. So now the other level of editing is like gene editing tools that affect the gene transcription level. So there are several of them. Some can introduce random deletions in the promoter regions. You can also create or disrupt the transcription factor binding sites in the promoter regions. For example, in the case of blast or blight, there are the bacteria tries to take over the host cell machinery by mutating the promoter regions where the proteins bind, the bacterial proteins bind and modulate and take over the plant host machinery. So those sites or specific base pairs can be edited through this so that the bacteria is dysfunctional and cannot affect the plant. Similarly, you can insert a fragment at the promoter region through non-homologous end joining or molybdenum template mediated knock-ins, and you can increase the expression of the downstream coding sequences. And also you can edit the epigenome via using of any demethylase or methylase domain along with the dead Cas9, which the dead Cas9 is not having any double strand activity, break activity, because both the domains are inactivated. The RUBC and the HNH domains are inactivated, but it still retains the binding capacity, the sequence recognition capacity it retains. So it can still recruit the demethylases or the demethylases or the methylases and allow the expression of the downstream or the inhibition of the expression of the downstream. So depending upon the domain that you've used, you get an eventual result. So this is how people have used the technology to see the out of box applications. Like you want to see where the genome sequence is actually, where the protein is localized or where the gene is localized. You just tag it with a fluorescent protein, and then you can see through this technology. And also you can overexpress or underexpress genes using this dCas9 platform. For example, if you want to overexpress a gene in the plant, and not using the transgenic approach, so the dCas9 approach can be used by utilizing the CRISPR-I or the CRISPR activation of the CRISPR inhibition. You can utilize a different domain. For example, for activation, you can use a VP64 domain along with the dCas9, and you can use a few guide RNAs of the promoter regions of the gene that you wish to overexpress. So those eventually can lead to the overexpression. For example, here they have tried to increase the purple, this pigment, anthocyanin pigment here in the purple roots. So through this technique, they have tried to increase the edited lines have shown this purple pigment here, anthocyanin pigment here in the roots. So it's like the other level, final level of editing tools is that work at the post-translational or the post-transcriptional or the translational level. So these are including several base editors that we can use to introduce or mutate the mRNA binding sites that interfere with the mRNA binding mediated regulation of different genes or mRNAs. So further, you can also introduce certain mutations in the donor and the acceptor sites and introduce missplicing or mRNA splicing effects, and thereby affect the expression of the downstream gene. And also, in the plants, it's been shown that if you knock out the upstream ORF, it leads to translational deregulation by overexpression of the downstream genes, and also some translation enhancers could be introduced to increase the translation of the genes eventually. So this is an example of how A to G mutation has been introduced in the FT protein, the flowering locus protein, as well as the flowering locus T protein and the PDS phytodesaturase gene. So here they've shown how this technology can be used to create neo-functional genes by introducing A to G substitutions or C to T substitutions by cytidine base editors. You can see the mutation in the flowering T locus. There is a delayed flowering trait that is been seen here, and introduction of this mutation in the PTS3 is led to the albino different percentage of expressions, like here 52%, 66%, if it's 87%, it becomes albino. So the anthocyanin pigment is totally bleached out. So this is how you can use this technique for opening up new avenues for plant genome engineering and biotechnology. So this is how in Arabidopsis VTC2 gene was mutated, and a similar gene in lettuce was also mutated. The GGP2 and the GGP1. These two genes are specifically important for ascorbate biosynthesis. So genome editing of these endogenous URFs have shown in these two plants that modulation can affect the translation of messenger RNAs, downstream messenger RNAs. So 4 important ORFs were overexpressed once this leader sequence in the mRNA was deleted. So this is how you could see the deletion of the upstream mutation in the upstream ORF led to the downstream overexpression of the downstream 4 genes that led to the increase in the ascorbate content, and also it increased in the oxidative stress reduction. This was tested by the paraquat that has been used. Paraquat is a kind of a reagent, as a herbicide which creates oxidative stress. So you could see that in these edited lines, the oxidative stress was less as compared to wild type. So the other important thing is a crucial prerequisite for a precise genome editing tool is basically the capacity to distinguish between the off-targets and the on-target sites. So precise methods for equitable genome editing, is based on the cognizance of the short guide RNA and the Cas9 protein, creating off-target double-strand breaks that lead to off-target indels, and it is really a very important point of concern for using these technologies on a wider scale and in the fields. So people are worried about this. So most of the guide RNA generation tools have been based on animal genomes, animal studies. So as of now, we still use those tools and screen the more effective guide RNAs. However, but eventually people would are developing newer tools that are based on plant genomes, and that would help us to create large scale data, numerical data, to improve our computational design of computational designing rules for developing or optimizing these guide RNAs to maximize the on-target effect and minimize the off-target effect, so that the experiment is more effective, the genetic screens are more effective. And another important problem with this technology is that there is a stringency of the PAM sequence. There are several enzymes that are there which have a stringent PAM sequence requirement. For example, in CPF1, TTTB, and so people are trying to create genomic variants with lesser PAM stringency or requirement of non-canonical PAM sites, so that this could help in creating more important mutations without the requirement of this specific sequence, which is not available most of the time, or sometimes it's less frequent in the genome. So to empower these, people have tried to create more relaxed PAM variants, and also these are promising for plant genome editing. Then the bacteriophages were, the viruses were, killed by the CRISPR-Cas9, but eventually to fight back this, the viruses have developed anti-CRISPRs. So these can also, be used to modulate the expression of Cas9 gene in the plants, because this would help us to regulate the Cas9 window, the window in which the Cas9 is effective, so that off targets are eventually, curbed. So there are various ways to do this. Several of them are, utilized to deactivate the site, and some are leading to dimerization of the Cas protein. So there are various ways, various anti-CRISPRs that have been now identified that can regulate the Cas9 expression. Then there are several techniques that have been used for introducing the editing of the mutation without introduction of the backbone, the vectors, and all. So this can be done through in vitro synthesis of ribonucleoprotein complex on the bench, and then shooting it with through ballistic gun or agrobacterium. So this is a more safer way, creating less off targets, because the Cas9 is shot as a protein and not as a gene. So this is also being utilized in plants, and it requires either a protoplast transfection method, or it can be used through directly introducing the RNP into the immature embryo, or the callus derived by immature embryo. So this can be done through a ballistic or through the protoplast transcription assays, and then they can be screened with PCR or restriction enzyme assays, and then these stable calli can be then regenerated, and the screens can be targeted to verify the mutations. So then there are other ways wherein you can contribute towards increased targeted sequence replacement within the cell, which is at a very low percentage naturally, by reducing the or removing the non-targeted or the error-prone mechanism of non-local end joining mechanism. So there are several inhibitors that used in this way, and also there are ways where you can create combinations of FOC1 along with the deactivated Cas9 for specificity, increase of the specificity. So this is how, this is how we create the CRISPR genome-wide screening libraries. And there are ways, like I told you, that you can fuse the inactive Cas9 domain to the fork1 nuclease to increase the specificity of the genome modification too. This is also one of the ways you can reduce the off targets. And develop newer delivery mechanisms is also important, and there are several already known. Recently, they've shown that if you transform the plant with a baby boom and a BUSTIL2 gene, it leads to the increase in the regeneration capacity and the transformation capacity, and it is genotype independent. So these mutants could have been developed through CRISPR, and they can be used for increased, regeneration and mutation capacity, transformation capacity. That would lead us to strategies that are genotype independent. Eventually, all the tissue culturists, crib about genotype specific dependencies of transformation. So that can be reduced through this. And also this nanotubes and nanoparticles and viral vector deliveries and tobacco rattle viruses, and all these are used to, because the virus plasmid does not integrate into the plant gene. So it can be used as a vehicle, and it is increases the amount of the total quantity of the DNA that is going in the plant. It increases that, the number increases, but it eventually the viral vector doesn't join in the doesn't it doesn't get introduced into the plant. It can be used safely. So all these techniques which I've spoken about, the genome knockout, the gene knockout, the gene replacement. Gene knockout is the loss and gain of function. I'm just summarizing this for you. The gene knock-in and replacement through homology donor templates, then the base editors, base editing approaches, then fine-tuning, that is the DCAS9 the DCAS9 platforms, then the DNA RNA virus resistance, as well as the high throughput mutant libraries. All these have been utilized for generation of different traits in the plants, like I previously said about abiotic stress, nutritional enhancement, as well as herbicide resistance and yield improvement. So there are several. We have developed, and the scientists all throughout the world have developed the CACO, which is resistant to a virus that was ravaging West Africa, and we would have lost our chocolate industry due to this virus. But thanks to the redeemer, CRISPR-Cas, as a redeemer, it came as a redeemer, and they could generate resistant varieties. So now we are happy with this. Then bananas and these grapes, they are also affected by a fungal disease that is very lethal. So the main important banana that is being used worldwide is under stress because of this fungus and also the grapes. So they have developed resistant varieties for that. And then they have also developed corn and rice varieties which produce more grains under stress conditions, drought conditions. And coffee, which has been decaffeinated, has been produced. Wheat, which has less gluten, has been produced. Mushrooms that are anti-browning, that are remain white all the time. The shelf life is increased, and tomatoes with critical flavors have been produced. So there are different other examples, as I've previously explained. And there are other examples. For example, lycopene-enriched tomato fruits have been introduced by knocking out the important 5 genes that convert the lycopene into alpha carotene, beta carotene. If you knock out these genes at this stage, the lycopene, the entire the GJP, that is starting material, will be converted to lycopene. It won't get further converted into alpha carotene, beta carotene. So you increase the lycopene content here. So this has been done. Then enhance the grain. Technology we use to enhance the grain yield in this maize by and knocking out these few genes there, which are mainly specific for increasing or repressing these genes, which increase the grain yield. So these have been used. The gene that has been already known in Rhabdophilus has been shown to affect the growth, the height of the plant, as well as the grain yield, seed yield. So they have tried to knock this gene to increase the seed yield and to reduce the height of the plant. So this is also how it increase the yield of the crop. Then there are wheat lines where they have used to increase the genes have been this the gene has been knocked and the cytokinin abnormal cytokinin response the one repressor gene has been knocked out to increase in nitrogen use efficiency. And there is this example where the both blast and blight resistance had been increased simultaneously by knocking out the BSRD1, the PI21, and the ERF922 simultaneously. So both blast and blight could be introduced in indica rice. Then there is an example where in the soybean, the isoflavone content was increased by knocking out these three genes that are common isoflavonoid substrate pathway genes, and it led to the increase in the isoflavone content, which led to the increase in the hairy root and the increase in the stress resistance to soybean mosaic virus. Then there's another example where carotenoid-rich rice has been created by knocking out a region where they say it's called the genome safe harbor. Genome safe harbors, they knocked out that, and from the chromosomal regions, and they introduced genes in the carotenoid pathway, which led to the increase of the beta carotene in the grains. So similarly, and the same, and then they also try to increase the stress resistance through knocking out this CCD carotenoid cleavage dehydrogenase 7 enzyme, which is creating the beta carotene into strigolactone. So by stopping the production of this strigolactone, they have tried to engineer the architecture of the plants. So they could get shorter plants, dwarf plants with higher yield. So they have tried to do this, and then they created aromatic maize. According to that, as I showed previously, they knocked out these two homologs. So in rice there is one gene there, BADH, and here there are two homologs, 2A and 2B. So they knocked out these two genes. So the GABA was not created. Entire content went towards the production of 2-acetylpyrroline, which creates the fragrance. They have done the fragrant maize seeds have been developed for creating popcorn. So then banana has been CRISPR has been used to edit the carotenoid pathway, where it has tried to knock out this epsilon cyclase gene, lycopene epsilon gene, which does not allow the lycopene to create alpha carotene and lutein, but the entire lycopene goes towards the production of beta carotene. So they have tried to increase beta carotene in the fruit of bananas. It's also, that beta carotene is a precursor of vitamin A. It's good for the eyes and other helps to fight night blindness. Then coffee. Here, this is a platform generated in coffee. They've tried to knock out this phytoin desaturase gene to show that now any other gene can also be introduced. So they've efficiently used this platform of CRISPR in coffee also. And here people have tried to increase the GABA content in the tomato fruits by knocking out of these 5 homologs. They've tried to knock out glutamate decarboxylase 3 and 2. These glutamate decarboxylase 2 and 3 involved in the GABA biosynthesis catabolism. So once they are knocked out, so they do not allow the glutamate to be converted to they allow this increased amount of GABA production, and this allows helps in increasing the resistance to heat stress in tomatoes. So another thing that I wanted to point out is an important one, where plant breeding could not help in creating this important trait. So what happened in the domestication of tomato? There were 100 years back, or more than 100 years back, there was a gene that was introduced through domestication. It's called EJ2, and which led to the increase in the calyx and the fruit size. And there was later, 70 years ago, there's another mutation that was introduced through domestication, which led to jointless pedicels. So when the farmers tried to, through breeding, tried to bring these two traits together in one crop, they could not find, if the interaction of these two alleles caused extreme inflorescence branching and sterility. So too much of branching they saw, and too less fruits produced, and the fruits also that were produced were sterile. So this was because of the negative epistatic effect that led to this. So this could not be, reverted or improved through crossbreeding or the normal conventional breeding processes. So what generally the farmers they used to use suppressors of this negative epistatic effect, but that suppressor utilization of those suppressors led to the reduction in the yield. So the Zachari's lab, Zachary Lipman's lab, in Cold Spring Harbor Laboratory, tried to generate different allelic variants of these two genes through CRISPR-Cas technology, and they identified the specific variant which has an adequate level of expression of these two to create an intermediary architecture which has weak branching and improved productivity. So this could have been achieved through the dosage from the selected alleles allows the improved inflorescence architecture and yield. So this is how the jointless and the larger calyx both together could have come into one through CRISPR-Cas. So this is how they did. It was just a repetition. And this is again the pictures of the paper. You can see how they've tried to improve the architecture. And also they've also tried they've tried to introduce these 5 mutations in 5 genes to introduce a specific structure and increase the size of the fruit, increase the number of fruits, and also the nutritional value, lycopene content. All this has been done by people. 500% lycopene content was increased. So this is how they've compared the pictures, and you can see at the lycopene there was 500% increase in the fruit. Also the size and the number. So what we have been doing in our lab, in short, I would want to finish. I've already taken now almost 2 hours, 1.5 hours. So I'm sorry for that, but I just wind up by a little bit of showing you all the work that we've done. So we've tried to use this CRISPR-Cas technology, which comes under the SDN1. So now the Indian government has allowed to test these genome-edited lines in the field, and they have categorized these into SDN1, SDN2, and SDN3 varieties categories. And the SDN1 is the simple knockout, which is like you are using the homologous, non-homologous end-joining pathway, and it is creating some insertions and deletions in the endogenous system on its own. So we are not introducing any insertions and deletions. The endogenous system is introducing that through the mutation and repair, and that is termed as SDN1, side-directed nuclease 1. And the other is SDN2, the side-directed nuclease 2, wherein you are introducing 2 or 3 mutations in the gene or the protein. So we've tried to develop these SDN2 lines in maize, rice, and pigeon pea lines by creating 3 mutations in the conserved region of the EPSPS enzyme, which is the 6th important enzyme in the shikimate acid pathway in the chloroplast of plants, which is important for the production of aromatic amino acids. So these aromatic amino acids are crucial for the plant growth. However, when the farmer sprays the herbicide, it affects this PEP binding. The PEP is the second substrate of this enzyme, phosphoenol pyruvate, which is inhibited by glyphosate, the herbicide Roundup Ready. So that inhibits the PEP binding. Instead, the glyphosate goes and binds to that region. So what we try to do is, like in the farmer's field, when the herbicide is sprayed, the rice field or the maize field or whatever, the weeds are killed because of this, because once you spray the herbicide, it deactivates the EPSPase enzyme, and hence no aromatic amino acids are produced and the weeds die. However, simultaneously, our main crop is also affected because our crop also has chloroplasts. Our main crop also has the same enzyme. So this our main crop is also affected. Tips are burned, browning of tips happen, and the shoot tip, the growth of the shoot tip stops. The plant looks young, but actually the old leaves remain green, but the young leaves start dying. So to protect that, to protect the yield of the main crop, we try to edit this PEP site. And I don't know whether this video would work, but I don't think so it will work. So it's trying to just show how the modification is being done and in the PEP binding site. And the glyphosate is unable to bind because the PEP, the binding site, active site becomes narrow and the glyphosate cannot bind to it. Hence, it is resistant. Our plants are resistant to glyphosate. So this is how we've tested it. This sequenced it. We've patented it. So these are the T1 lines that are grown in the tissue culture medium. The edited lines are growing happily in 4 millimolar glyphosate as compared to the control plant, the treated control plant. And then we verified the lines with Cas9 gene, and we also checked the sequence of the PSPS. We gave it for sequencing. We verified it through sequencing. Then we developed homozygous lines with the removal of the Cas9 gene, and we sprayed these lines with glyphosate up to 6 ml per liter, and we see that our edited lines are happily growing in comparison to the control plants when they're sprayed. They cannot recover back, but they recover back in our fields. So these are there in our greenhouse. This is a small greenhouse that we have for where we have grown this, and the photosynthetic parameters were checked in comparison to the treated control. Our edited lines performed very well, and the EPS-based assay activity was also done through malachite dye-based assay, and you can see in compared to the treated control, our lines have active EPSPS. The shikimic acid is the first the substrate of the HPLC. The pathway. The first substrate is the shikimic acid, and it is eventually utilized perfectly in the edited lines as compared to the untreated control. Whereas the treated control, the activity of the EPS space is reduced, so it will not use the shikimic acid. Similarly, we checked the end product, the aromatic amino acids. So compared to the treated control, our edited lines are showing increased amount of it's not increased, but significantly increased amount of phenylalanine, tyrosine, and tryptophan, which are the aromatic amino acids, the end product of the biosynthetic pathway. So this is another picture where comparative morphology has been seen with respect to the wild type. You see there are no defects in the size of the cobs. The kernels are perfect. There are no edge dislocations. There are no missed kernels. There are no developmental defects. So they are very parasynthetic. So this has been done in the rice also. And just skipping the slides just to show you the main parts. So in the rice, the edited lines, there's a Bangladeshi student, Sonia, who developed these lines. So there are these were tested with the strip acid and the glyphosate, and they showed resistance to glyphosate. And then here we've seen that in the media also, the edited lines and comparison to treated control, they are growing perfectly fine, and they were sprayed. And there's well recovery in these edited lines with respect to the wild type. The recovery is faster and the yield is not compromised at all. And they showed significantly improved aromatic amino acid content as compared to wild type. And also we developed certain rice lines with increased iron and zinc content in the endosperm, which is definitely 2 to 4 ppm normally, but we managed up to 15 to 20 ppm. So I would wanted to show you the way we did it. We just wanted to show you, like, this is the way we predicted it before, because we wanted to show that the proof of concept. We tried to create models to quantify the iron and zinc levels in the rice grains by creating a total network of the rice genome. You take all sequence data or whatever mutational data, synonymous, gene expression profile, protein interaction, whatever data available publicly or through in-house data, we just used it to develop this model for taking these genes, the 3 genes that we picked for knocking out these were basically iron sensors, and they were trying to create they used to create a feedback loop to and sending a signal to the roots to stop absorbing iron and zinc. So when we knocked these sensors out, there was no feedback signal going to the roots to stop the iron and zinc to be transported to the grains. Hence, there was an increased accumulation of iron and zinc in the endosperm of the grains. And this is how we use these genes as their nodes, and they took them as the node naturally was selected as the least neighborhood centrality, and it was done so that the influence of these three genes when they knock out is less on the entire network. So we found this is how it looked, and when we knocked these three genes out, there was homeostasis that was created in this system. It was not affected. This is the wild type model where you can see these three genes, and you can see these two genes here, the nicotinamide synthase and the ferritin genes. These are the two important genes that people have tried to overexpress in the past in the transgenic plants to increase iron and zinc, but they have failed terribly. By the simple experiment of knocking out these three genes, we could increase not only these two genes in the endosperm, but around 18 to 27 genes that are related to iron and zinc uptake, transport, and storage. So once you see these enhance expression of these genes that are related to iron, zinc uptake, transport, and storage was observed. So this was then ranked according to the matrix, and we saw that the ranking matrix, the two important genes that I talked about that were used for developing transgenics previously for increasing iron and zinc in the endosperm of rice, in the white rice, I'm saying, not in the brown rice. So that is these are the two target genes that were always used for experiments. So and this is how we converted the entire expression profiling data in the in-house data. We converted the gene expression data according to the formula here. It is known as the intensity-based absolute quantification. We converted it into ppm, and we found that it would increase up to 15 to 30 ppm, but eventually I have figures, but I'm not showing them because we have yet to patent this. So these are the lines that are there in the greenhouse. Khalid from Egypt developed these lines and for his PhD, pre-doctoral fellowship, arthrophilasty pre-doctoral fellowship. And these are the lines. So these have increased iron and zinc up to 60 ppm. So we're trying to develop allelic variants that have the optimum iron content so that it is absorbed by the human body. But we're trying to do that in the mice. We're trying to test those in the mice. We've identified the allelic variants that are best expressed. So that we have also trying to develop low phytate cereals and tomatoes with reduced allergenicity and increased vitamin D and vitamin E. And this is my team. I thank you all for the patience. I am finding this up, the names of these students that have contributed to this work, and there are two more students that have joined, but they have not. I'm not included the pictures. They have recently joined, Pooja Chakravarti and Isha Gupta, and there is a postdoc, Ram Kumar, who has also joined us. So all of them I thank, and I thank the audience sitting there, and I'm very happy to take any questions from the audience. I'll be glad to answer if there is. Thank you so much.
Thank you so much, Dr. Tanushri. It was really elaborated information on the genome editing. We do not have any questions from here. Thank you so much for your time.
Thank you.